Okay, we're gonna continue with our lessons. The next thing we're gonna learn in terms of biotechnology is DNA fingerprinting. Right. To understand DNA fingerprinting, we have to understand something called polymorphism. Polymorphism is basically any differences in the DNA that can be detected. Okay, so like if there's some kind of DNA difference between you and I, if there's a way to detect it, then we call it a polymorphism. And these polymorphisms, these differences can occur either in the coding or non-coding regions. They could exist in the exons or the introns. And the kind of differences that you observe comes in different forms. So one type of polymorphisms that we are interested in exist in these regions called short tandem repeats. Okay, so tandem is one after another. Short is 2 to 3 nucleotides, and they are repeated over and over again. So short tandem repeats, we call them STR. For example, over here, we have CAG, CAG repeated 4 times. So we write it as CAG4. Right? That's a short tandem repeat. Right? So the polymorphism exists between individuals in the sense that you will have CAG perhaps repeated 5 times. I might have CAG repeated 6, 7 times, right? So the repeats are the same, but the number of repeats might be different between individuals. And that's what we're trying to detect when we do DNA fingerprinting. Now, sometimes it is possible for these short tandem repeats to cause problems. Okay, one example of this is Huntington disease. Huntington disease is a neurodegenerative disease where a person lose control of their motor skills and they tend to wobble quite a bit. They have uncontrolled movement of their limbs, not to be confused with Parkinson. In Huntington disease, what happens is the CAG is repeated, okay, repeated more than 36 times. Right? Normal individuals who do not have Huntington only have CAG repeated 10 to 35 times, but anything beyond 25 times, then the person will have Huntington. Typically, the disease don't start to show symptoms until after around age 40, and by then, most people would have kids already. This is a dominant mutation. So if one of the parents have Huntington disease, there's a 50% chance that the child will have it, right? And that places a person in a precarious situation. Like they could do genetic testing to find out, but there's a 50% chance that they're going to have the disease, right? Some people don't want to know. Some people want to find out, and it's somewhat of a dilemma in most instances. But not all STRs are linked to disease, and for the regions where the STRs are not connected to any kind of conditions, it will be useful to use them as a way to distinguish DNA samples from one person to another. Right? So for example, person number 1, they have AGAT repeated 12 times, but another person, they only have it repeated 5 times, right? Some STR might be the same, and then others are going to be different. So the key thing to take away here is to understand everybody has these short tandem repeats. You have it, I have it, and then they are located at the same chromosomes. So if I find AGAT on chromosome 17, everybody will have it on chromosome 17. What's different is that the number of AGAT might not be necessarily the same between you and I. Okay. People who are closely related, they might have more STRs with the same number of repeats compared to people who are not related. So what we can do next is to basically analyze the short tandem repeats from different people based on how long the repeats are. And to separate DNA, based on the length, we have to do something called gel electrophoresis. Okay. It's much easier if I show you the video, the clips of how it looks like, rather than me explaining it. Okay. I mean, like I'll be explaining it to you, but I'm going to be showing you the video as well. So this is a typical setup for gel electrophoresis. We have these little tubes that I've been telling you about. They are called Eppendorf tubes. The name is not important. That's where you will keep your DNA sample. So here we have 1, 2, 3, 4, 5 different samples. These are pipettes that you would use to pick up the DNA. And then this is a power supply that will supply current to separate the DNA. Let's take a look at what they're going to do. This is the reaction chamber. There is a negative terminal, which is black, and a positive terminal, which is red. Okay. And so in order for this to work, what they're going to do is they will create a gel. Okay? The gel is kind of like how you would make jello. Okay, so it comes in a powder form. You reconstitute it in a little bit of liquid, and then you wait for it to set. Okay, and the gel looks kind of like this mishmash type of structure. And there is the gel right there. It looks very smooth when you look at it like that. But like I said, the actual gel structure, if you take a closer look at it under the microscope, they are these little mishmash. Okay, so for the DNA to move through the gel, depends on the size. If it's big, then it's gonna get tangled easily. It's not gonna be able to move through it quickly. But if it's really short, then it's able to weave through the gel easily. Okay, so as an example, I can show you this. So that's the gel, right? With the slots at the top for you to load the DNA, as you would see. And this is what the zoom in version of the gel looks like. It's like the honeycomb structure, right? So it's kind of like this thing, play structure that I saw at the playground with my kids. So they challenged me to get up at the top, get in at the top, and then see who can get down to the bottom the fastest. So they are really tiny compared to me. So they were able to move through it very quickly. They can, get through the gaps and everything. By the time they get to the bottom, I was still stuck, midway in the structure. And that's kind of what's happening with the gel electrophoresis. When you have a sample of DNA, some are long and some are short. The longer DNA fragments will have a hard time weaving through the gel, just like I have a hard time weaving through these obstacles. Okay. But if you're small, you can make it through quicker. That's the main principle of gel electrophoresis that allows you to separate the DNA based on the size. Now, let's go back to the gel over here and see what they're going to do. Can I speed this up? Well, anyways, okay. So they are pouring in. Yeah, I saw, but it's okay. It's sleeping. Yeah, so they're gonna pour in the buffers, okay, which is just to speed up, which is just to conduct the electric current. Right, so remember, each of these tubes contains a little bit of DNA sample. And
Sorry, they're putting gel into the water.
The gel is in the buffers. Yeah, it's not water. It's like a solution. There you go. Yeah. Okay, so this is the pipette, and yeah. Okay, so look, right? That's the DNA sample right there. Right? It's not a lot. It doesn't look like a lot, but already you have thousands and thousands of copies of DNA in there. Right? And the DNA is not supposed to be blue. The blue is actually a dye that you add to your DNA sample. Right? The dye has two functions. And write it down when I tell you what it is. Okay, just watch first. So they're gonna suck up the DNA sample mixed with the dye. There you go. And then they are gonna basically load the sample into the gel. There are those slots at the top, right? So that's one sample right here. That's another sample. The dye, the blue dye, helps increase the density of your sample so that when you inject it into the gel, it's gonna sink into the well. If it's not dense, more dense than the solution, when you add DNA, it's just gonna squirt all over the place, and you will lose the DNA sample. Right. So function number one of the dye is to increase the density of the sample so that it will sink into the well. Okay, so it will sink into the gel. So here's another thing that I probably should explain to you. This is the top view of the gel. And again, you have these slots at the top. Right? And this is where you're going to load the DNA sample, right? So the electric current goes from negative to positive because the backbone of DNA is negatively charged. It's sugar phosphate. The phosphate is PO4 3 negative. And so if you apply current from negative to positive, it will push the DNA down the gel. Okay? And each of these lane is for each of your sample. So if you load the sample here, the DNA will travel down the gel this way. It's not gonna go diagonal, all right? It's just gonna stay in its own lane. You understand? Right? So that's one lane, second lane, so on and so forth. Right? And over here, they're gonna load another gel, another lane. Okay. Goes into the well, and so on and so forth. Once it's ready, once you blow all the lanes, you close the lid, and you turn on the power supply. Okay, and then you can see, when you see the bubbles, you know the current is being conducted in the solution, in the buffer. Okay. And as the current is going from black to red, it again pushes the DNA along the gel. So this is, again, looking at it from the top, and the blue dye kind of gives you a rough idea of where the DNA samples are. So you can see it's being pushed from the negative terminal towards the positive terminal. Okay, so this is, as time goes on, you see the sample moving down. So the second function of the gel, of the dye, is to give you a visual way to track where your DNA is. Right? You can't see the DNA here. Okay? The DNA is like invisible, right? It's like colorless. But the dye gives you an idea of roughly where they might be at. Right? So if you see the dye is here, it means the shorter DNA is probably somewhere here. And then the longer ones are probably here. So when you do the experiment, you don't want to do it for too long. If the blue line goes all the way to here, you might have lose some samples already. Okay, it might have escaped from the bottom of the gel. All right. So how do you visualize the actual DNA? Well, to see the actual DNA, you actually need to have a special stain called ethidium bromide. And these things will bind to the DNA and cause it to glow, to fluoresce when you shine UV light at them. And then you can take a picture of the gel. Okay, so that's what it looks like under UV light. Each of these bands corresponds to a DNA. Okay, so in this sample, for example, you have two pieces of DNA of different length. Okay, and over here you have many. So this is another picture. You can see only one DNA sample here, one DNA length in this particular sample, and so on and so forth. So I want to ask you, which one is shorter? Is this one in number 2 shorter, or this one in number 4 shorter? 4 is shorter. Anyone else? 2 is shorter. Yeah. Okay. So remember, this is one lane. This is another lane, right? And they are both racing to the bottom. Which one gets to the bottom easier? The shorter one or the longer one? Shorter one. So number 2 is actually shorter because it's able to move further down the gel in the same amount of time compared to 4, right? If the further down it is from the gel, the shorter it is going to be. Now, you can't really see this too well because of the lighting, but do you see multiple bands here? Yeah. So that actually is called a ladder. Ladder. And a ladder, or sometimes called a ruler. A ruler. DNA ruler, DNA ladder. And what that does is these are prefabricated DNA of known lengths that you can buy from online. If someone made these DNA and they made it to be a certain size. So when you load it along with your sample, it gives you a reference to check to see how long your DNAs are. So you see a band here, then you can compare it with the ladder. You see a band here, you can compare it with the ladder. That way, it gives you an idea of the size of your sample. Let me show you an example, okay? So let's take a look here. So we have a ladder here in lane 1, and then this is our sample, and we see that our sample contains how many DNA fragments? 3, right? Right. So these 3 fragments were created by cutting it with restriction enzyme. Right, so we had an original DNA, we cut it twice, which gives me 3 fragments. Okay? And the question is asking you, what's the length of the original DNA sample? Okay, so let's take a look. This one, compared to the ladder, right, what size do you think this is? Huh?
9.
9, right? It's between 8 and 10. So that one is 9, right? 9 kilo base pair. 9 kilo base pair.
That's pretty cool, but.
1,000 base. 1,000 ACTGs. Okay. Yeah. What about this one? How long is this? 6. Yeah, I would say it's 6, right? Okay. That's it. The thickness doesn't really matter. Okay, it just means you have a lot of it, basically. And this one?
2.3.
2.5, yeah? 2.5 sounds good. So when you add them all up, what do you get? Yeah, so the original DNA before you cut it is 17.5. You understand? Yeah. I know it's hard to focus after the test, but please focus because this will be on the test next week. Okay. There is the review package. Okay, if you haven't picked one up, make sure you pick one up. I will take this up next Tuesday. Okay, next Tuesday is gonna be online. Remember? Right? Next Tuesday is gonna be online.
Oh, it
You
is?
didn't know? Yes, because I said you guys have the chemistry test in the morning, right?
Yeah.
Did you guys do a chemistry test on Monday? On
No.
Tuesday? Yeah.
Yeah.
So then, I'm gonna do the review for you online instead, okay? So that you can, rest up after your chemistry class or
Oh,
whatever.
so are you gonna record it?
Yes, yes, yes. I'll
Okay.
record it, yeah.
Okay.
Why are you guys so shocked? I told you guys this before, and you guys were like happy when I first told you, and now you're like, what's going on? Okay. We have class this Thursday though, okay? Come to class this Thursday, okay? It's the Tuesday when I take this up, right? That's gonna be online. My goodness. Huh? You want it to be in person? Do you guys actually want it to be in person? No? No? Yeah. Yeah. Okay. Well, yeah. I think traditionally I do it online is better. Okay. Yeah. Okay, let's do another one here. We have what do you call this thing? Huh? What is it called, sorry? No, it's in the question, guys. Plasmid. Yeah, it's a plasmid. Okay. The plasmid are circular DNA. And over here, we are treating the plasmid with equal R1 and BAMH1. Okay, we're going to treat the plasmid with these 2 restriction enzymes. And then afterwards, I'm going to load the products onto a gel. How many bands are you expecting to see on the gel? Good. It's not rocket science. Okay, just think about it. Right? We're going to treat it with these 2 restriction enzymes. Good. So where is it gonna cut?
Um,
Yeah, it's gonna cut where it has the same name, right? So it's gonna cut right here and also right here. And then that's one fragment, one piece, two pieces. Okay? I also want to ask you which piece will be further down the gel? The one on the right or the one on the left?
One on the right.
right.
Because it's shorter, right?
Yeah.
Okay. So that's pretty straightforward. Okay, read this and tell me the answer. Yes, good. Yes. Okay. CAG repeated 10 times. How long is that? No. CAG 10 times. How long? 30. Does anybody not understand why it's 30? Just look at me if you. Wait, 3 times 10 is 30, right?
Yeah.
Okay. How about this one? How long is this? 45. That's it. 45. So is 45 longer or 30? 45. Which means this is going to go further, right? The answer is A. Understand?
Wait, which means, wait, sorry, can you repeat that again?
Sure.
What if thought that it would be the second one because it's longer, so we'll reach.
Because it's longer, it's not able to move through the gel as easily, right? That's why it's like me who's stuck
Yeah,
at the
yeah,
top,
yeah.
right?
Oh, okay.
Yeah. All right, we're gonna do a quick note. Do you guys have at least 3 colors? All right, here we go.
This is gonna be a long.
Like a 10-minute note. Where do people use DNA fingerprinting? Have you guys heard of DNA fingerprinting? Forensics, yeah. So if you want to determine if the suspect is at the crime scene, right? That's DNA fingerprinting. What else can we use DNA fingerprinting for? When you go to jail? What's that? Biometrics, sure. Anything else?
Your phone.
Huh? Your phone?
Like, that's
I
ID.
don't think, no. Not we're not there yet, yeah. To figure out who the father is, right? Sometimes, right? Or like, if people are siblings, so on and so forth, right? Do you guys know, like I told you about 23andMe, right? They they recently went bankrupt. And what happened is, as part of their liquidation, they are actually selling all the genetic data that they've collected to another company, right? So even though they say, we're going to safeguard your information, they're not really doing that anymore. And basically what happens is to find out whether you have like some long lost second cousins or something, they are basically using DNA fingerprinting to match it
I
with the
thought it
pool.
was spit though.
Hmm?
I thought it was.
There are many out there, right? There is yeah. Anyway, so I'm gonna show you how DNA fingerprinting works. Okay, so let's have a person here, okay? We're gonna name this person Bob, okay? My favorite name. So over here, let's say we have chromosome 3, chromosome 7, and chromosome 14, let's say. Now I'm gonna use different colors here to help you understand. Let's say over here, Bob has an STR that goes like this, ACC repeated 10 times, okay? And over here, he has another one, CG, repeated 5 times. And then on chromosome 14, there's another one, AGC, repeated, oh, I don't even know, 15 times. Okay. So these are short tandem repeats, okay? You have it, I have it. We can look up where they are using the genome browser that I showed you last time. Okay, so let's have another person. Okay, this is gonna be, I don't know, Amy. And Amy, who's also human, would have the same STR. But the repeats might be different, right? I keep on telling you. So maybe she has 20 repeats instead. And here is going to be CG 10. And over here, ACG 5, okay? What?
He doesn't have.
Oh, sorry. Thank you. Yeah, long day. Very observant, yeah. All right. Now, we know where these STRs are because, again, we have the Human Genome Project data. And so what happens is you can design primers to only clone those STR. And so we can do like a cheek swap from Bob and Amy. That would have their entire genome, but then I would only supply primers for this region, and this region. Remember, primers are like the sticky tags, right? Okay, so you tell the PCR where to copy. You're not going to copy the entire genome. We're just going to copy these interested STR. Okay, and they're going to be at the same location. So what you're going to end up having is you're going to have one of those tubes. And in the tubes, you're going to have the, millions of copies of the red STR, blue ones, and then the green ones as well. Okay, so PCR will clone all 3 STR regions. Okay, same thing on the other side. And we're going to have a gel. Remember, the gel is negative at the top and positive at the bottom. I'm going to put it in one of those chambers. You're going to run a current. We're going to separate these things. I'll give you a moment to draw it out. And I'll tell you what it looks like. You have to draw gels on the test, okay? Final test. So make sure you understand what's going on here. All right. It's not too bad. Just stay with me, okay? And stop me if you don't understand. How long is this? 30, right? 3 times 10 is 30. How long is this one? Yeah, 3 times 20 is 60. Do we understand how to get the numbers? Okay. How about this one? 2 times 5 is 10. This one? Yeah. 45. 15. Okay. So we have all the numbers here. Let's take a look. Which one, remember, the entire sample from Bob, let's put it right here, is going to have 3 fragments. Which one is going to be the furthest down in the gel? Red, green, or blue? Red, green, or blue? Green? Because green is the shortest, right? So it's going to be all the way down maybe here. Okay, again, in real life, there are no colors. You can't see them. It's just like a band, right? Like the pictures I show you. I'm only using the color to help you understand. And then which one will be closest to the top, the blue or the red? The blue. Blue is 45, which is longer than 30, right? Okay, so I'm going to put it around maybe here, 45. And then the red is 30. So it's not going to be like all the way down here. I'm going to say it's roughly here. So take a moment and draw Amy's band. All right, we're gonna load Amy's sample. We're gonna load the sample right here. He also has three fragments, three STRs. Where will they be? You got it? Which one is closest to the top for Amy? Because it's 60, right? In fact, 60 is even longer than 45. So it has to be higher up. You understand? Okay. It's going to be higher up than the 45. And then 20 and 15, they are pretty far down, but not as far as the 10. So I would put 20 probably around here. And then 15 is slightly shorter. So it's going to be really close. So with just 3 STR, we are able to generate a unique almost barcode for each person, right? And we can tell who's who. But in reality, we cannot just use 3 STR because people who share same ethnicity, for example, they would be close enough that 3 STR is not enough to distinguish them. Okay, so what we actually do is we do it for many STRs, and then you end up getting something like this. So like at the crime scene, we have these are STRs, right? Like the ones we did, right? But just with many more. So that's the crime scene, and then we have 3 suspects. So who was there? Yeah, number 2, right? Yeah, it's exactly the same. Understand? That doesn't mean he, was the person who did the crime, but he sure was at the scene. Okay. All right. Forget this. It's not on the test. We don't have time to go through it. All right, let's take a look at this one. This one is fun to do, sort of, maybe. We have a family here, right? 2 parents and 2 kids. Who are the parents and who are the kids? Right, so these are STRs, right? Okay. Remember, all your genetic information, half of it you get from mom and half of it you get from dad, right? So let's take a look at the first one. Let's take a look at the first one. First one says B is the child of A and C. B is the child of A and C. Is that possible? So let's take a look at B here. Right? So if B is the kid of A and C, that means everything B has came from either A or C. So let's check. Okay. Where did B get this one from? Okay, got it from C. So this one is good. Okay, got it from C. Where did B get this one from? C. That's good. And where did B get this one from? Oh, no, we're looking at like A and C, right?
Oh.
Yeah. Where did he get it from? No one. That means A and C cannot be the parents of B because B has something that neither of the parents have. Do you understand? Oh, good. I never seen so many heads nodding at the same time. Let's try the next one. And do you think C is the child of A and B then?
No. No.
Is it yes? Yeah. No, who said no? No? Why not?
Oh, yeah.
The fourth one? Okay. So 1, 2, 3, 4, right? The fourth one, where did C get this one from? From A, right? It's okay. So keep checking. Is there something that's unaccounted for? Yes, there is, okay?
No, yeah, it is.
Yeah, it's good, right? Everything is accounted for, right?
Yeah.
So if these are the only choices, then yeah, they are the parents. Understand? So this is wrong, okay? So this is correct. This is correct. Okay, can we try the next one, please? It's much easier if you do it on paper, okay? Huh? Did they print on page?
Yeah, I didn't know this.
Oh, that's okay. Yes. Yes, Ruth, that's right. Yeah. Anyone else? Mm-hmm. Yeah. Okay, so let's take a closer look again. Is the first one correct? D is the child of A and C.
Oh.
So let's see. This one came from here. This one is okay. Okay, okay, okay. Okay, okay. This one's not okay, right? The fourth one is not okay. 1, 2, 3, 4. Oh yeah, fourth one's not okay either, right? Okay, so that means this is no. Okay, if you find something that didn't come from A or C, not the kid. All right, what about D is the child of A and B? All right, I think if you check everything, all of these can come from A or B. So that's the answer. Okay, so that means A and B are parents. And so who are the siblings? C and D. Okay. That's it. So that's how you can use DNA fingerprinting, right? This is, of course, an oversimplification, but that's how you know in real life. All right. Good. We are right on time. So for the last part, the hard stuff is over, guys. Right? The last part is quite straightforward. All right. We're gonna talk about gene therapy briefly. So gene therapy is, you know what? Actually, let me just double check here. Okay. Gene therapy is the idea of introducing genetic materials into a cell. If you introduce the genetic material into the somatic cell, which is all the cells except for the ones that will become sperm and egg, and it's called somatic gene therapy. Somatic gene therapy will only affect the person who's receiving the gene therapy. It's not going to get passed on to future generations. That's not the same if you do germline gene therapy. Germline gene therapy will affect the gametes, it will affect the sperm and the egg, and it could potentially have multi-generation alteration. Okay, and because of that, germline therapy is considered as unethical in many jurisdictions because you are altering the genes of someone who hasn't been born yet. Okay, so most of the research are focused on somatic gene therapy. Now, what are we trying to do here? The goal of gene therapy can be separated into three categories. Number one, replace a non-functional gene. If something is not working, you give the functional copy with no mutation. That should fix the problem. You can try to cover up an overly active gene, or you can change how the disease progresses by introducing certain kind of genes. Let's take a closer look at each one of these. The first one is most straightforward. Okay? Let's say someone has sickle cell anemia. Remember we talked about sickle cell anemia?
Yeah.
It's a missense mutation. So the missense mutation causes people to make misfolded hemoglobin, right? And that creates the disease. And so logically, we can deliver the normal functional version of the gene, the one without the missense, and it goes into the cell, then you will be able to make the normal hemoglobin, and then you're going to be able to fix the disease. Right? This typically only works if the condition is recessive, okay? Because if the condition is dominant, it doesn't matter how many good copies you supply, the dominant mutation will cover all of them up. That's the meaning of dominant, right? Dominant means you only need one copy for it to show up. So that's why it wouldn't work for dominant condition.
How long people live with sickle cell anemia?
How long?
How long, in average, people?
Oh, people don't die from sickle cell anemia, but it's a condition that causes them to have very poor quality of life. Okay, so there's a lot of pain. They have shortness of breath. It's not something that would kill them, but something that is not pleasant to live with.
In case they are dominant, how long they can live?
It would not in the case of sickle cell anemia. Okay, sickle cell anemia is a recessive condition. You have to have both. Yeah. Now, you also have to understand when we use gene therapy to try to fix some of these problems, if the condition already caused some irreversible damage along the way, treating it with gene therapy is not going to undo those damage. Okay, let's say there's some kind of mutation that causes developmental problems along the way, and the person receives treatment at age 10, right? Well, the first 10 years of damage cannot be undone, right? It will slow the progression or stop the progression, but you cannot undo the harm, right? So some disease that has irreversible damage is not an ideal candidate for gene therapy, not at a later age anyway. Okay, so besides sickle cell anemia, there is something called beta thalassemia. This is caused by a nonsense mutation. A whole chunk of the hemoglobin is gone. This is actually pretty severe. The person needs to be on constant blood transfusion to survive. And with constant blood transfusion, they actually get iron overload, and that could cause other problems as well. So it's a good candidate for gene therapy. And I want to mention that gene therapy is not something in theory anymore. There are successful cases where sickle cell anemia and beta thalassemia has been treated with gene therapy. Okay, but the technology is not very mature and popular yet. That's why it's not widely available. Okay, we talked about hemophilia, which is a bleeding condition. So these things don't have lasting effects, generally speaking, and they make it ideal as a candidate for gene therapy. But not everything can be fixed with replacing it with a normal functional copy. So another goal is to suppress an overly active gene. We learned about overly active genes before in cancer, right? The green light that doesn't turn off, for example, right? That's an oncogene, right? So if you have an oncogene, if you have a protein, that causes the cell to keep on dividing and dividing and dividing. What you can do is you can insert a blocking gene. So the blocking gene, it's exactly what it sounds like. Okay, it covers up the overly active gene. Right? So think of the green light that's always on, right? Well, what can we do? Well, we can put a cover over it so that we don't see it anymore, and that could, stop the effects of the overly active gene, and the cell hopefully will return to behaving normal. Finally, we also have a third goal, which is to alter the clinical course of condition. And typically, we're talking about cancer once again. Okay, and there are two ways to do it. You can deliver what's called a suicide gene. The suicide gene will go into the cancer cell, and then the suicide gene will get turned on, and it will make some kind of toxic product that causes the cell to die, which is ideal, right? If the cancer cell is able to kill themselves off because of the suicide gene, then the disease would be under control. And sometimes, instead of doing a suicide gene, you can insert a marker gene. One of the reasons why it is so difficult to treat cancer and for your immune cells to kill the cancer cell is because cancer cells arise from your own cells. So they look very similar to your own cell. Your immune cells is unable to tell the difference between what's normal, what's healthy, and what's cancerous. Okay? And what this gene therapy aims to do is they will introduce a marker gene that goes into the cancer cell, and that would cause the cancer cells to put special signals on the surface. So when a white cell come and see these special signals, they would attack it. They would know, hey, that glowing thing on the surface of the cell, that's not supposed to be in the body. Let's go destroy it. Okay. So this one relies on the cancer cell killing themselves. This one puts a target on the cancer cells so that your white cells can destroy them.
Which one is more effective?
Right now, this is still very early stage. I think they have some success with the marker gene. But when we go through some of the challenges, you will understand why it's not as easy as it seems. Now, there are two ways to approach this gene therapy thing, okay? You can do it outside of the body, which is called ex vivo, or you can do it inside the body, which is in vivo. In ex vivo, you would take the cell from the person, out of them, and then you would manipulate them, you would change them, modify them in the lab. And afterwards, you make sure that all changes are good, and then you put it back into the person and let it do its job. And so this ex vivo method is a little bit, you have more control over it, right? Because you're only doing this pool of cells, right? You can make sure that it's completely functional before putting it back into the person. In vivo involves using some vector, like a virus, to help deliver the gene into the person. So a little bit less control or as to like where the gene is going to go, but it's more cost effective in many cases. What are the key challenges that are facing that we're facing with gene therapy? So targeted delivery. This is a big thing. Right? How, like for example, if you want to deliver the suicide gene to the cancer, how do you make sure the suicide gene only goes into the cancer cells and not the healthy cells? Because if it goes to the healthy cells, then they start killing the healthy cells as well, right? So that's difficult to control. And immune response. Sometimes, like if you do the marker gene, yes, you will trigger the immune system to attack the cancer cells, but sometimes if you overly excite your immune system, they could do weird things and they could start attacking your own cells as well. I recently read a book and it was describing like a real case of a person receiving gene therapy for skin cancer, right? So when he was receiving treatment, the skin cancer was regressing, right? But then a couple months into the treatment, he started having liver problems because the immune cells got influenced, if you would, and they start attacking his own liver. Right? So he has to come off of the gene therapy, and then the liver got better, but then the cancer came back. So there's a trade-off between the efficacy and the immune response.
Oh, is that why, like, when people have, like, are doing chemotherapy, they, like, because it can't target to this certain, like, just specific cancer cells, it makes you sick?
Yeah, in a way, right? Chemotherapy targets rapidly dividing cells, right? So it's not just the cancer cell that's rapidly dividing, right? Your hair cells, for example,
Yeah.
right? That's why people lose hair and then
Yeah.
their bone marrow source. Okay, interaction with other genes. What if the gene, instead of just covering up the overly active gene, it covers up other genes as well, right? It's really hard to predict exactly what's going to happen until you run with the experiment. That's why a lot of these things are still in clinical trial phase, but there are some encouraging data. And recently, maybe past 4 or 5 years, there is a new technology called CRISPR. Some of you might have heard of it. And CRISPR allows people to very precisely decide where to put the gene in. They could tell, go to chromosome 17, this position, take out this much, and then put in this much. Okay, and this technology is, I think, is going to revolutionize the gene therapy in the next 10, 20 years. This is going to be big, and it's going to be able to cure a lot of diseases which we currently don't have a cure. But the most concerning thing right now is cost, right? There was, do you guys know the show, like America's Got Talent? Yeah.
Yeah.
There was a couple, like more than 10 years ago, I think. There was a contestant. He was very good at singing, but he was blind. Okay. And so I think he won, and then that got him a lot of fame. And his kind of blindness is actually a congenital. He was born with it. And it turns out he was missing a protein in the eyes that caused him to not be able to see, right? So the gene therapy is perfect for this, right? So he raised awareness, and when you're famous, you get people funding you, right? And he was able to get gene therapy, and he was able to see afterwards. Okay, you can look up this person. But what was the cost? The initial cost was 60. No, let me get this right. I usually get the numbers wrong because it's different in English and Cantonese. $600,000 US for one eye. Okay, and subsequently you have to keep on paying 100,000 every year to keep it going, because it's not something that you can fix in one shot, right? So I mean, like, who has the money to do that, right? Certainly not the average individual, and it's not something that insurance is likely to pay for, right? So cost, socioeconomical barriers, all these things are going to be a big challenge. And that is all for today, guys. Very good.